Hello.
Hi.
Hello.
Good morning.
Velkommen.
Welcome.
Welcome to UEG Week Virtual 2021.
Whatever your level of expertise, you will find the best and latest updates on UEG Week Virtual.
I wish you those days full of interesting, diverse and creative content.
I count on your contribution and I thank you.
Viel Freude, ingest the best.
Ingest the best.
Just the best.
Ingest the best.
Ingest the best.
Key word は ingest the best です。
Get ready to ingest the best.
Ingest the best.
Ingest the best.
Well, good evening, friends. Welcome back to our virtual studio here in Vienna. We hope you have been enjoying and ingesting the best of UEG Week Virtual 2021, where we have over 8,500 registrants and 106 countries, people from all over the world exploring GI. I hope to have, again, your participation in this session throughout the Q&A tool. This is our UEG Research Prize session, the most prestigious prize that we give out at UEG. We're also going to be hearing from two late-breaking abstracts, so lots to get through. Let's welcome our in-studio faculty. They are Professor Luigi Ricciardello, the chair of the UEG Research Committee, the UEG treasurer, Professor Bertold Kolecko, Professor Rebecca Fitzgerald, who's from our UEG Week faculty, and joining us from Germany, Professor Dirk Haller, our Research Prize winner. Very welcome, Dirk. It's great to have you here. A shame you couldn't join us in studio.
Yeah, it's fantastic to join finally.
Great.
So I'm grounded at the moment for a COVID infection, but you know, this is just a minor detail here. I'm absolutely flattered to be in this session to get this prize.
Well, it's great to have you, and I'm so sorry, and I hope you're not feeling too bad, but it's great. It's an important session to have you, so thanks for your time. Thanks also to Jennifer Seely-Voigtner and Leanne Heuthorst, who are going to be presenting their late-breaking abstracts. You're both very welcome.
Thanks. Thank you.
So, Jennifer, I know you're joining us from the United States, so thanks very much for joining us. What time is it there?
Well, I'm actually on holiday. It's noon.
Okay. so you've just gotten out of bed.
Yeah.
Well, thanks very much. Looking forward to hearing your extract. But Bertold, as chair of this very important session, can you tell us a little bit about what we can expect?
Well, as you said already, we'll hear two late-breaking abstract presentations. I'm really looking forward to that, and I hope we have a lot of good questions from the audience. And then, of course, we will award our most prestigious prize, the renowned UEG Research Award.
You have obviously been involved with UEG for quite some time. Have you been preparing for this week? Have you been looking before the week to some of the abstracts and some of the e-poster exhibitions?
Well, the UEG Week each year, of course, is a big highlight. There's so much exchange of new information, great research, good educational updates. And in the executive committee, of course, we are busy preparing for many months for it, not as much as the office team is, but we are still involved, and we can't wait to see the abstracts that are accepted and peek at the abstracts, and are excited about the work that we see.
Well, I suppose that's one of the things you get to do in your position. So, Rebecca, you're very welcome as well. I'm looking forward to chatting to you in a bit. But let's go straight, I think, to our first late-breaking abstract. It is from Jennifer Seely-Voigtner from Newport Beach in the United States. And we're going to discuss this now. So if you have questions for Jennifer, please do put them into our Q&A tool, and we'll get to them as quickly as we can. The talk is Celiac Shield Gluten Challenge Study. Glutenase protects the mucosa and attenuates symptoms in celiac disease patients. We'll hear the talk and do questions after.
Many thanks to UEG for allowing me to present these excellent results from our latest phase 2b clinical trial. supporting safety, tolerability, and efficacy of lataglutenase, a drug candidate in late phase 2 clinical program for the treatment of celiac disease. Here are our disclosures. Lataglutenase is a clinically proven non-systemic orally administered biologic enzyme treatment that degrades gluten in the stomach. Lataglutenase is meant to be taken as an adjunct to a strict gluten-free diet. The enzymes are in powder form and should be dissolved in water and then consumed with a meal. The Celiac Shield study was a National Institutes of Health funded study, specifically by the National Center for Complementary and Integrative Health, was a double-blind, placebo-controlled gluten challenge study of the efficacy and safety and tolerability of 6 weeks treatment of lataglutenase in patients with well-controlled celiac disease. It was a single-center US-based study done at the Mayo Clinic in Rochester, Minnesota. 50 patients were randomized, 43 successfully completed the study, despite problems with COVID and recruiting. Here are some histologic data summarized here. The 2 plots on the left are change in delta VHCD and IELs from baseline, which of course are the cornerstones of histologic measurement. Blue is lataglutenase and red is placebo. From the plots, you can see significant attenuation of mucosal damage by lataglutenase. The plot on the right shows results translated to the Marsh-Oberhuber scale, which the FDA still likes to see. Same conclusions stand, statistically significant histologic protection. We also evaluated histology by several other methods in order to cross-correlate our findings. But the same data shows attenuation of mucosal damage was demonstrated with high statistical significance. Here are the results from a secondary, more exploratory endpoint that got us very excited. Measuring gluten immunogenic peptides in urine measures the effectiveness of lataglutenase mechanism of action, which is degrading gluten in the stomach. Here is a preliminary experiment looking at LC-MS and a lateral flow assay showing the basic retention profile of gluten in urine. In the study, we used the lateral flow assay to measure gluten GIPs in urine. The V3 and V4 points are baseline measurements before and following the gluten challenge treatment. As you can see, these levels rise abruptly during the gluten challenge for placebos, but barely change for the lataglutenase subjects. These results show individual patient trends, and you can see there's a distinct difference between placebo and lataglutenase. The left plot shows individual patient changes, and the right is a scatter plot showing the actual change for each patient. Looking at symptom data, the green and orange bars show symptom severity levels for baseline and gluten challenge for lataglutenase versus placebo. You can see statistically that there is significantly more worsening for placebo and lataglutenase for several of these symptoms. Here are some trends analysis that shows the symptom worsening by 3 2-week periods. The trend is highly statistically significant as evidenced by the p-values. Here is some data for measurements of gluten-induced antibody levels via serology. Each of the 3 titer measurements showed worsening for placebos, shown in red. However, surprisingly, the lataglutenase subjects in blue did not show attenuation, but basically got better despite being on a gluten-free challenge. In summary, we achieved clinical and statistical significance in all 4 endpoints: histology, symptoms, gluten, and GIPs, and serology, demonstrating lataglutenase is an effective drug. I'm grateful to be able to represent a great many people who contributed to making this study an overwhelming success. On behalf of everyone involved, I thank UEG and the audience here today for your attention.
Oh, I can see why we had this paper, a really great paper. Thank you very much for that, Jennifer. Bertold, over to you.
Yeah, thank you, Jennifer, from my side as well. That was really a great presentation. While we are waiting for the audience to type their questions, maybe I can start. I noted that you had, there's a previous larger study with a different dose with 900 milligrams of the 2 proteases. So can you perhaps comment what was the trigger to do this new study with a larger dose and a smaller patient number?
Well, we're getting ready for trying to decide how we're going to go commercial. And so we saw dose dependence from 100 up to 900 milligrams. And we wanted to make sure that the dose dependence continued above 900 milligrams. So we chose 1200. So we may go to commercial launch in phase 3 with 600 as well as 900, as well as 1200 milligrams.
The other question I would have is, it's great to see the difference between placebo and control, but from a clinical point of view and probably the point of view of the patient, the question also is how many of the patients would have a true success, for example, villus to crypt ratio beyond 2. Can you share that as well?
Well, again, we are confident that we can show a bigger healing factor in our phase 3 study when we have a longer trial duration. So the fact that we could see healing in such a short period of time is very exciting. And so in our phase 3 study, we will no doubt have a bigger difference in the healing that you'll see from villus height crypt up.
Rebecca, you have a question as well?
Yeah, I mean, it follows on from that, exactly. So what do you think the duration of the dosing would require to be? Do you think this is something patients would have to stay on continuously? And what might the side effect profile of that be?
Well, so far, this has been the most studied therapy to date in celiac trials, and the safety data has been impeccable, mostly because it's a non-systemic therapy. Yes. And so the treatment will be ongoing. It's not a cure. It's meant to be an adjunct to a strict gluten-free diet. And so the patients would take it if they think, for example, at a restaurant or a party where they think they may have gluten contamination.
Thanks.
Luigi?
Yeah, I have a couple of questions. The first is whether you have any information about malabsorption, if you have checked, data on other outcomes in these patients. And the other question that I have is, what was the starting point in terms of disease activity, where all the patients march Oberuber, 3B, 3C to start with?
Yes, patients in the study were on a gluten-free diet for over a year. So they were not at their worst as far as, a very, very sick and diseased intestine. So as far as malabsorption goes, that hasn't been studied in this particular study because, again, the patients were already gluten-free for over a year. So we have done previous studies with newly diagnosed patients. So we haven't seen any issues with malabsorption of the drug in those patients.
Great. Thank you very much, Jennifer. That was really insightful. Let's go on then.
Thanks very much for that, Jennifer. Our next paper, Rebecca, is on CRC, and I know this is one of your areas of expertise. What do you make of the UEG oncology program this year?
Yeah, well, oncology obviously is a huge part of gastroenterology and what we do. And we've seen a really fantastic, diverse program in oncology across a number of different cancer type areas. Today, I particularly enjoyed the oral presentations around advanced screening methodologies for screening colonoscopy, a lot about artificial intelligence. And of course, the take home there that artificial intelligence is only as good as the endoscopist and the clinician, still treating the patient. So I think that's an important thing that resonated with me, and I think we need to bear in mind.
How have you enjoyed your day? Any highlights from today's activities?
Yeah, and then also I really enjoyed a treatment on advanced therapies for early upper GI neoplasia. Incredible the sort of advanced therapies we have in our armamentarium now and the lengths you can go to try and still preserve the organ for the patient, which I think is a really, really major advance.
Great. Okay, let's hear Leanne's talk then. If you have questions, please put them through in the Q&A tool. This is Leanne Hothurst from Amsterdam in the Netherlands. Her talk is Increased Rate of Colorectal Cancer in Patients with Ulcerative Colitis, a National Cohort Study between 1991 and 2020.
I have no disclosures. I would like to thank you for the opportunity to present our work. My name is Lianne Heuthorst. I am a PhD candidate at the Department of Surgery of the Amsterdam University Medical Center. And today I would like to present the results of our population-based study on the colorectal cancer rate in patients with ulcerative colitis over the last 3 decades. So patients with ulcerative colitis are known to have an increased risk of developing colorectal cancer, which is related to the pro-neoplastic effects of chronic inflammation. However, the impact of this effect remains debated as data on the colorectal cancer risk in UC patients are conflicting. The first meta-analysis published in 2001 demonstrated a cumulative risk of colorectal cancer of 2% at 10 years, 8% at 20 years, and 18% at 30 years in UC patients. In following meta-analysis, including population-based studies, lower colorectal cancer risk rates were found. So yeah, the colorectal cancer rate might decrease because of the expanding medical options we have these days, leading to improved control of mucosal inflammation, severe mucosal inflammation, and also because of the implementation of surveillance programs. However, a key question in this discussion is, does have the current treatment strategies, do they have a true modifying effect in the natural history of the disease, or does it merely delay colectomy? Because if indeed current treatment strategies lead to postponed colectomy, this might increase the risk of colon cancer in the long term. So the aim of our study was to identify a time trend in increasing disease duration before resection, accompanied by a shift in colectomy indication from therapy refractory disease to malignant degeneration over the last 30 years. So we performed a retrospective population-based study using the Dutch pathology registry. The Dutch pathology registry covers all the hospitals in the Netherlands, and from this registry, all pathology excerpts from patients with histologically confirmed UC undergoing a colorectal resection for all indications were included, and the primary outcome was the colorectal cancer rate in the colectomy specimen, and outcomes were compared between 3 time periods: period 1, period 2, and period 3. So if we look at the results, in total 6,094 patients with UC were included, of which 5,026 underwent a proctocolectomy and 1,068 a segmental resection. And the CRC of the colorectal cancer rate was 16.9% after a median disease duration of 11 years. In this slide, in the table in the first line, we see the overall number of resections per time period. And in the second line, we see the number of patients with colorectal cancer. Here, if we look at the proportions in red, we see surgery for colorectal cancer, and we see an increase from 11% to 16% to almost 23%. And in green, we see the proportion of surgery for all other indications. So we see a significant increase over time. And in this table, we look at the clinical and pathological characteristics of UC patients that underwent a resection for cancer. And we do see that the mean age increased over the years from 57 years to 61 to 63 years. And if we look at the disease duration, this increased. This is the median disease duration, which increased from 4 years to 10 years to 17 years. And if we look further to the pathological staging, we see that the majority of patients was diagnosed with a T4 tumor, over 60%, in all 3 time periods, which did not significantly decrease over time. And if we look at the positive lymph nodes, which was 33% in the first period, 42% in the second, and 38% in the third period. So the amount of the proportion of patients with positive lymph nodes did also not decrease over time. So in conclusion, we observed a shift in surgical indication from therapy refractory disease to colorectal cancer. Patients underwent resection at an older age and after a longer disease duration. And we hypothesized that the expanding therapeutic options might postpone colectomy, which might increase the risk of colorectal cancer in the long term. And unfortunately, the incidence of advanced colon carcinoma remains high, despite the implementation of surveillance programs. I would like to thank you for your attention, and I would love to answer your questions.
Thanks very much for that, Leanne. Fantastic paper. Rebecca, questions?
Yeah, thank you, Leanne. I really enjoyed that. And these kind of studies, these epidemiological studies, are so important to kind of understand the changes over time. And I just wondered, are there other registries that you can look at to see if this is a more widespread effect that's being observed elsewhere, or is it something particular in the Netherlands? I'm sure not, but I'd be interested to know if there's any other data we could look at to help us understand how general the problem is.
Are you aiming for registries in the Netherlands, or do you mean more globally in Europe?
Both. Are there any other data to say whether this is observed beyond just in your centre?
Yes, I think there are another few publications demonstrating that there is an indication shift going on from therapy refractory disease to cancer. So I think we're not the only country or only center observing this, but it's definitely something we should look further into.
And then, of course, there's the kind of understanding what's underlying this. So, I mean, you kind of put out some hypotheses at the beginning. You know, on the one hand, if we're controlling the disease better and controlling the inflammation, that might be expected to reduce the rate. On the other hand, if we're waiting longer and the natural history is the same, that might actually increase the rate. I suppose there's also the possibility that the medication we're using and immunosuppressive effects may actually be a pro-carcinogenic factor as well. Do you think that's relevant or not?
Yes, and next step, I think it's really important to know, to find out who these patients are. So to identify the risk factors, to know if what kind of severity of disease they have, the extent of disease. So next step will be to look in our own cohort together with another center to find out the exact risk factors and details about these patients, so we can better select them and identify them and find out what kind of surveillance programs we need to detect precancerous lesions in an early stage.
Yeah, absolutely.
We've
got some questions now coming through from our audience, which is great. So the question from Maria Gonzalez Vivo: do you have data about dysplasia or CRC that were found in the pathology results in surgeries for other indications?
So what we did is we just we selected all patients with UC who underwent a colorectal resection, all indications. So, and then we scored if they had a colorectal cancer in the pathology report. So in this cohort, the patients with colorectal cancer are also patients who just underwent surgery for therapy refractory disease.
Okay, thanks. There's a comment on the chat here that there's also consortium data about colorectal cancer risk in IBD in the Johns Hopkins study. recently, which looked retrospectively at data from 1991, so that might be a good source. And I've got another question from a colleague here.
Yes, first I'd like to add my applause to this great work as well. But in relation to this question that Rebecca just mentioned, I heard two fantastic presentations this morning from the group in Lille in France, who looked at a cohort of 1,300 plus children with IBD over a period of 25 years. And on the one hand, they reported, of course, an increased malignoma risk long-term in these children with IBD. But the other interesting observation was that over the 25 years, there was a marked improvement in the outcome. For example, 1/3 less intestinal resection, 1/2 less colectomy in the UC children with the advent of course, improved treatment strategies, which of course is great to see that implementation of better standards can improve the life of these children. But my question really is, what should we expect from your point of view with the lesser colonic resections long-term in these kids?
Oh, yeah, it's a really good question. Yeah, I think it's wonderful if you can prevent a resection. I think it does a lot on the quality of life. But I think we really need to find out a good way to survey this patient so that we can so we can prevent that these patients develop cancer or are detected when it is in an advanced stage. I think that's really important.
Yeah. No, I completely agree. Early detection is key. You can't, on the one hand, improve quality of life and reduce colectomy if you then still have an advanced cancer down the line. So clearly we need to do better there. Thanks, Leanne. Great presentation.
Well, thanks very much for that, Leanne, and thanks as well to Jennifer. So it's now time for the highlight of the evening, the awarding of the UEG Research Prize. So Luigi, maybe you might tell me a little bit about this prize, what it recognizes and how you identify your winners.
So each year UEG rewards the best of the best in research, basic research, translational, clinical research. And the award is of 100,000 euros. And it's basically not just an award that is given to the researcher, it is actually funding a research project. So it's a 3-year research project, and so the researcher who is going to receive the award has to deliver, a research project with final data that hopefully are going to be presented at UEG Week. So what we do have within our research committee, we have we are valuing, the proposals, and I can tell you that this is very competitive because the selection is made upon the quality of the research and the outstanding track of the researcher. Of course, we are looking for researchers who are really active in their research career. We are not awarding, researchers who are in the late stage of their career because, of course, they need to deliver a research project.
How does this differ to funding from other sources like European or national funding, this particular prize? What's the benefit of winning this particular award for the awardee?
Well, I think it's important because it provides the awardee is tied to UEG, and UEG recognizes its excellence among the peers that are working within the digestive health research community. And I think it's also very important for the young researchers because then gives you, the perspective of our research community being awarded by UEG that is really recognizing the best of the best.
Can you give me a little bit more detail on what you look for from applicants?
Okay, so we judge actually 50% the quality of the proponent, and so we look for independence in research, the track record, of course, if she or he has been well funded by the the funding bodies that we have in Europe, and then 50% goes into the details of the research program. So it has to be highly innovative. It has to go with a proof of concept, and it has to deliver from start to the end something that is going to be a breakthrough in the respective field.
Which is exactly what Rebecca delivered as a previous winner of this award. Can you tell me what winning that award meant for your lab and for your research?
Yeah, I still remember actually the enormous buzz and honour when I heard that I got that award. It was a huge thing, and the excitement of giving the presentation. I'm sorry that you haven't got a chance to give that presentation live, but I just remember still the kind of that nervous energy of going up and delivering a talk to a large audience. And you've got a large audience today. But also, although you have to deliver on a specific project, there's actually a lot of flexibility in how you, in the end, choose to spend the money to deliver on that project, which is quite different actually from many other grants organisations who are kind of breathing down your neck, and you have to spell out to the last euro exactly how you're going to spend spend it. So here you're given flexibility. You have to say what your idea is, how you're going to deliver it, and then you're left to get on with it. And you know that UEG are genuinely believing in you and interested in what you go on and do with it. So fantastic.
Brilliant. Luigi, we're going. I'm teasing this out. We will award, but just before we do, can you tell me what about this year's winner stood out for you?
So actually, that was a very thorough selection that we had to make, because there was a huge display of wonderful researchers. And this was a very innovative project, and it was unanimously judged as the best. And it's very interesting because the awardee is actually repurposing some drugs that are for treating other diseases, like mitochondrial diseases, for as an adjuvant treatment for inflammatory bowel diseases. And it goes with a very sophisticated approach, because the project will be delivered using organoid cultures coming from IBD patients, from mouse models. And so we took into consideration all the advancements that he's going to provide with this technically challenging project, which I think is going to be very interesting.
Well, you teed it up very nicely. Ladies and gentlemen, for this year's project, Repurposing Mitochondria-Protective Targets for Adjuvant IBD Therapy, please give a warm round of UEG applause for this year's Research Prize awardee, Professor Dirk Haller from Germany. Such a shame you couldn't be here. We have our research officer here, Matilda, who I know you've been dealing with already. We'll be dealing with the prize. So this is your research prize. It's a beautifully designed prize, if I say so.
I can see it. Yeah, it's fantastic. Looks great. I take it.
Congratulations, Dirk. We'll send Matilda off on her bicycle now, and she will knock on your door in a few days' time. Thanks very much for that, Matilda. Dirk, how does it feel to be this year's UEG Research Prize winner?
Well, it's an exceptional moment. I think for a basic scientist, it's probably even a bit more exceptional and flattering, because you're in this medical research community, and we're trying to do translational research, I think, since I started my career. And to get this kind of appreciation from your peers is a wonderful moment, and probably means, at least to some extent, that the research we do seems to be clinically relevant. And I think that's probably what research at the end should be relevant. And we're trying all our bests along this way, and to get this kind of appreciation, I think is just fantastic. It feels great.
No, it's great. You have the full support of UEG. You have €100,000, and while it's not exactly a blank check to do whatever you want with it, as Rebecca said, it is different in that it does give you a great amount of freedom. Did you recognise the potential in this project, and did you feel that this actually has a chance of winning?
Well, you always hope, huh? Otherwise you wouldn't submit a proposal. But at the end, you're then struck that you get it, because it really means that to some extent you hit the target. When I look at this project, it is really something which were working in my lab more than 10 years. And it's really also describing my background. It's I'm in a very multidisciplinary environment. I'm a nutrition scientist dealing with microbiome in the gut. And at some point, metabolism is very close. And getting this idea that, a disturbed metabolic tissue as a residual disease is still influencing the outcomes of inflammation, relapse, and so on, or the way medication is actually working in these tissues. So I think this bridge between metabolism, inflammation, and the way we see tissue healing, I think is something very close to what we're trying to achieve. And it's actually fantastic that other people think that this is a way to go.
Dirk, briefly, is there anyone you'd like to thank for the award?
There's many people involved, but I think there's actually 2 points I want to make. One is the type of research I do depends on clinical collaborations. And within the European network, I think we established friendships, we established great collaborations with clinician scientists. They trust us, that we deal with their samples, which are precious, in the right way. So especially I want to highlight two, which in the last 5 years at least gave us a lot of valuable samples. One is Julien Panis in Barcelona, the other one is Mathieu Allais in Paris. And especially Mathieu in Paris will be instrumental also for this type of work to give us patient material. And the second point I want to make is the great lab, because at the end of the day, if you don't have young people in your lab which are driven in terms of drilling this down to results, this will never fly. So I'm happy to have this lab, and I'm happy to have these type of collaborations in Europe, and I'm very thankful.
Well, congratulations again. Let's hear your talk, and then we'll get questions from our online audience and from our faculty. The talk is on repurposing mitochondria-protective targets for adjuvant IBD therapy.
My name is Dirk Haller from the Technical University of Munich, and I'm very proud to receive the UEG Research Prize this year. The target is metabolism and reshaping of metabolism, and to use drugs which have been designed to treat metabolic related diseases as an adjuvant IBD therapy. I have a declaration of interest shown here. I'm participating in seminars, advisory boards, and have some co-funding for projects from industry. So the core aspect of this project is that the intestinal epithelium, which lines our digestive tract, is a single cell interface, and it is in close interaction with very complex microbial diverse and chemical diverse tissue milieu. And the question is how this interface is regulated. And it's absolutely surprising that on a huge surface, 30 to 40 square meters, we renew this single cell layer every 3 to 5 days. We have a great gradient from small to large in intestine with respect to the epithelial surface. So it's probably different between small and large intestine, how certain drugs also would interfere with epithelial functionality. So we worked over the last decades on the question how this tissue milieu, which is complex, as I said, is integrated into epithelial functionality. And we came across cellular metabolism and signaling pathways associated with the regulation of cellular metabolism. And we basically saw that this metabolic aspects of the epithelium is linked to functionality in the stem cell niche, renewal, is associated with proliferative controls and differentiation of epithelial cells. The question is now, if the cellular metabolism is rendered, is that part of IBD pathogenesis, and even is part of cancer development. So mitochondria and mitochondrial perturbation is a very important aspect of cellular metabolism. And mitochondria are basically, if they're disturbed, they elicit signals. And that's the mitochondrial stress response. It could be acute or chronic. And we propose that these chronic aspects of mitochondrial perturbation induce metabolic injury in the tissue. And one key proposition is that this injury, if it's not resolved at the metabolic level, would cause chronic tissue injury, which would then affect the chance in IBD, for example, that patients relapse. So here, the proposition that unresolved tissue injury based on metabolic disturbance would affect the recurrent disease behavior illustrated here. We have flares of inflammation over time. The big problem is that associated with this inflammatory activation, we always have tissue damage. We have the tissue renders. And it's certainly different of whether we talk about Crohn's disease or ulcerative colitis. Illustrated here from mouse models, from the basic science part, which we work very heavily on it. We generated different mouse models on the question of mitochondrial stress signaling, and one of those is the deletion of HSP60, which is the mitochondrial stress chaperone. And you can delete this mitochondrial stress chaperone specifically, for example, in all epithelial cells or just in intestinal stem cells. What happens if you delete it is that you elicit a signal response here. And this unfolded protein response from disturbed mitochondria, if you delete HSP60, is illustrated here. We have a hallmark transcription factor activating transcription factor 5. You see strong activation here in response to this deletion of the chaperone. But what's also very important is that you render cellular metabolism. So you basically reduce respiration, oxidative phosphorylation, and you increase glycolysis. So this shift in cellular metabolism basically occurs at the same time as these signal events. What you see here is a specific deletion of HSP60 in stem cells. And if you do so, then either the signal specifically or the change in cellular metabolism reduces stemness, stained here for LGR5 as the hallmark marker for stem cells. But what it also does in the stem cell niche, it renders functionality of associated cells, Punnett cells, and you see the granularity, how it disappears here when you disrupt this mitochondrial integrity and if you change cellular metabolism. Very important to translate this into the clinical setting is always the ability to get tissue section. And here I'd like to highlight my long-standing collaborator, Mathieu Allais in Paris, and he gave us tissue sections from a Crohn's disease cohort which underwent surgery. And the ileal resection and the generation of this near-terminal ileum was looked at time point at 0 at surgery, M0, and then six months later. And what you see is that this newly generated tissue early on can already have signs of inflammation or not. And here we looked specifically at those tissue sections at M0 which had no signs of inflammation. And still, these patients have a 50-50 chance to relapse. And without clear prediction marker to actually decide of whether or not the patients undergo recurrence. What we then looked at the stem compartment, and you can clearly see those patients which had a low chance of disease recurrence had an intact stem compartment, stained here for LGR5 in the patients, and those which had low stemness here had a high chance of disease recurrence. So this is the translation from the mouse models basically into the clinical setting. From the mouse model, again, you can mimic this. This is a Crohn's disease-like mouse model, TNF-delta mice. You see a gradient of inflammation in these mice. And what you see associated with, if the mice are strongly inflamed, you have almost a complete disruption here of the stem compartment and a massive reduction of LGR5 positive stem cells. What we did is we generated organoids, and these organoids from the inflamed mouse, they don't really live long. But if we expose those organoids with a drug which affects metabolism, it's an FDA drug approved, dichloroacetate, and it renders the conversion of pyruvate to acetyl-CoA. So it shifts basically glycolysis back into oxidative phosphorylation and respiration. And what you see here in analogy, what I showed before, is that you can basically rescue organoids. They grow much better ex vivo. So we can rescue the phenotype of stemness and growth ex vivo. And this is a very important proof of concept experiment to show that metabolic drugs affect signs of inflammation, which have to do with tissue regeneration. And this is basically at the core of this UEG Research Prize project, that we aim to look at metabolic drugs which have been approved already, used in metabolic diseases, and we want to apply them to organoids generated from patients, from mouse models, select some of those and go into the mouse. And we selected the drugs based on their function. So improvement of cellular respiration, for example, impact on cell and oxidative stress mechanisms, approval of mitochondrial structure and biogenesis. And we want to see of whether we can antagonize with these drugs signs of metabolic injury at the molecular and tissue level. And lastly, we want to use basically stools from patients which are in remission. And the question is, are in the stool supernatant metabolites, which basically would again have a regenerative phenotype and antagonize metabolic injury to improve tissue responses. That's the core of the project. And for this, it's very important to work together with clinicians. And I'm very proud that I have a long-standing great relationship with Mathieu Allais in Paris. He's very central to provide patient material. And I have a fantastic team in Munich, which basically takes this tissue and puts this in the perspective of molecular and cellular work we do in mouse models. Having said that, again, I'm proud to receive this prize. I'm very eager to work on this project. Thank you very much.
Well, Dirk, really exciting work, and thanks so much for telling us all about it. Bertold, questions?
Well, I think we really selected the right winner. This is all what a good research project should have: innovative hypotheses based on really good quality work, state-of-the-art methodology, and feasibility of trying to test the hypothesis with a strong team and a strong research background. So in my role of the treasurer, my role is to spend money for the right purposes.
Yeah.
It's a great pleasure to sign the check for such a great project.
Very good.
I'm sure I'm going to spend it, Bertold. No problem.
Okay, great.
Any questions from our faculty?
Dirk, what do you think is going to be the most challenging part of your project?
Well, at the moment, still, this is a little blue sky research idea, because for many of those drugs which we have selected and already generated a kind of a smaller number of drugs which we want to test, we actually have no idea how they would act on epithelial cells, for example. For example, take metformin. This is a standard drug to treat insulin resistance in diabetic patients. Still, the drug is surprisingly volatile and affects microbial ecology. It affects other cells in different ways. So actually, I really don't know what we are going to see. And that means that at the beginning, we probably have to go over some hurdles of frustration, need to screen, select some of the putative targets, which we then want to narrow down and follow up in the more complicated experimental setups. But I think that's the normal way of research. We are prepared for that, and we're facing it.
Any other questions from faculty?
Yeah, so it's really lovely to see that you're going to use a combination of the 3D organoid models and the mouse models, and you've got access to patient tissues, which is critical. So I just wondered the extent to which, and maybe this is beyond your project, but you can then actually examine the metabolome of your patients with disease or without disease.
You know, this is a good question. I mean, the chemical diversity of what we find in the gut lumen is enormous, and we just have, I think, scratched at the surface. And repurposing the drugs is actually, and probably the easier way to start. And it is a start because we know already that these drugs and these chemical structures have already a function on mitochondria, for example. But the long vision of that certainly is to expand on the drugs. And the good thing on the drugs is that you can use them as an adjuvant therapeutic approach. They're approved for safety. They may be easily embedded in an adjuvant strategy for treating patients. But on the long term, I think it is about knowing the chemical diversity and the functional diversity of metabolites in the gut lumen. And they come from metabolized drugs. They come from all sorts of microbial metabolism. They come from host metabolites. So this is a vast diversity which we want to explore and use the knowledge which we gain here, the technologies, the experimental designs to further implement these larger questions.
Dirk, can I ask how you felt when you found out that you had won? Where were you?
Where were I? You know, I had the enormous privilege to have a research sabbatical over the summer in Lisbon. So I felt terribly bad because in Germany they were suffering from bad weather. They had COVID. Well, we had COVID too, and I get it now, so it's probably fair. But I got this message, with 26 degrees of sunshine and having a lot of time to think about my work and to write proposals. So yes, I felt great. I was at a great place, and it was in Lisbon in my sabbatical, actually.
Very good. Well, look, on behalf of all of UEG, congratulations on a fantastic research project. Our Research Prize winner 2021, Dirk Haller.
Thanks so much.
Thank you, Dirk, and thank you to our remote faculty, Leanne and Jennifer. Thanks to our on-site faculty, Luigi Berthold and Rebecca. If you want to watch back any of the sessions, you can do so just by pressing the Missed a Section button on the platform, and those are all available until November 15th, and then they go into the UEG library where you can access them forever. If you'd like to join us in a few minutes' time, we will be giving out the best paper awardee from the journal. We'll be joined by Professor Joost Rent and talking about the UEG journal as well. So stay with us. In about 5 or 6 minutes, we'll be back here in studio with UEG Week tonight. We'll be back in a bit.